In this lesson, we'll learn about the genetic code. This is the language for living things on Earth. And this is a visual display of the genetic code. We appear to have these letters in the middle, and then around the wheel we see these abbreviations for the 20 different amino acids that are found in proteins. Now the letters, some of them look familiar. A and G and C, they sound like letters you'd find in DNA, but there's a U here. So this display is not the language of DNA, it's the language of RNA. Now, for the purposes of this lesson, I'll assume that you've already watched the animation about how cells access DNA information, make a temporary copy in a molecule called messenger RNA, and it's the messenger RNA that hooks up to the protein construction factory called a ribosome. So the ribosome chugs along 3 letters at a time, linking amino acids together into a protein. The animation I'm talking about looks something like this. Here we have RNA polymerase, and now the DNA has unzipped and unwound, and RNA polymerase now is going to be making a complementary messenger RNA molecule. If we just pause it for a quick second, we'll see that when there's a T in DNA, there's an A in RNA, but when there's an A in DNA, it's a U that sticks to the A. So messenger RNA has C's and A's and G's, but it has a U instead of a T. Now, the messenger RNA molecule is similar to a DNA molecule. It's a chain molecule, but notice DNA is a double-stranded molecule, whereas messenger RNA is just a single-stranded molecule, and it has that U that sticks to the A. So what the cell is doing here is it's making a temporary copy. The messenger RNA molecule is a temporary copy of the information stored in DNA, because the sequence of messenger RNA letters is identical to the sequence of one of the strands of DNA. So here we see just a still picture of the situation. We have DNA here, the double helix of DNA, it's all twisted up. But when the cell needs to make a protein, it's going to have to access the recipe for that protein. Proteins will come along and separate the 2 DNA strands, and then RNA polymerase will come along and build a messenger RNA molecule that is complementary to the template strand here, which means it's identical to the opposite strand. So let's confirm that. Messenger RNA here is the chain molecule, and it has a sequence of letters here. The G is sticky with C, the A is sticky with T, the C is sticky with G, the U is sticky with A. So messenger RNA does not have a T. The G sticks with the C, and so on. Now notice then the messenger RNA is complementary to this strand of DNA, but it's identical to this strand of DNA, with the exception of a U wherever there was a T. A U here, T there, U there, T there, UU, TT. So messenger RNA is a copy of the DNA sequence. It's got the recipe to build a protein, but it's going to be mobile. It's going to detach from the DNA and move out to where a ribosome is. And that's what we saw then in this picture. We have the messenger RNA has already hooked up to the ribosome here, and the ribosome is using other carrier molecules called transfer RNA molecules to bring the appropriate amino acids according to the sequence of letters in messenger RNA. A certain sequence of amino acids will be stuck together to form a protein. Okay, so let's remind ourselves that these abbreviations on the outer part of the wheel are the abbreviations for the 20 different amino acids. And the letters here inside the wheel are the messenger RNA letters. And so we've got the inner wheel here, we've got the middle wheel of letters, and we've got the outer wheel of letters. And they correspond then to the positions of a triplet. So in the inner wheel, we have the messenger RNA letter that is in the first position of a triplet. And in the middle wheel, we have the second position, and the outer wheel then is going to be the third position in the messenger RNA triplet. Notice then we also have 3 triplets that can represent stop. It means the end of the message. And in fact, the first triplet in a messenger RNA sequence is going to specify methionine. That's an amino acid. So now let's see how we can use this diagram to decode the amino acid sequence when given a messenger RNA sequence. So we're going to use this messenger RNA molecule right here, and you see it is a picture of the ribosome here, and we've got the carrier molecules bringing amino acids, and here 2 amino acids, they're going to be chemically bonded together, and we have the growing chain of amino acids, which is the growing protein. So let's use this mRNA right here to practice using the code. Well, the first triplet then here that we see is CGU. So we start in the inner wheel and we find the C quadrant. Then we go in the middle wheel, we find the G, so that's over here. And then it's a U as the third position, so it's this one right here. So I have a CGU. Now the outer part of the diagram tells me that's arginine. And so we have arginine as the first amino acid coded for by this mRNA recipe. The next triplet is GAC. We start inside, find the G, A, C. That's going to be aspartate. And then it's going to be AGU. AGU. That's serine. So the third amino acid is serine. Then we have CGU. CGU. We have arginine. Then we have ACG. ACG. That's threonine. And then we have UCA. UCA. That's serine. And then we have GAU. GAU. That's aspartate again. So this would go on until we get a stop triplet. So one of these 3 triplets would be at the end of every messenger RNA. So it could be a UAA is a stop signal. There's no amino acid. It just tells the ribosome end of the message. Could be a UAG or it could be a UGA. So each messenger RNA has a sort of a start triplet. That's usually methionine, and it has one of these 3 stop triplets. Let's use this diagram for more practice. So here we have the messenger RNA molecule, and we have spelled out the sequence of messenger RNA letters here. So we have an AUG. AUG is methionine, and notice the first amino acid is methionine. Then we're going to have a CGC. CGC, that's arginine. Next one is arginine. Then we're going to have, let's see, an AGC. AGC is going to be serine. The third amino acid is serine. Then we're going to have UUA. UUA, that's going to be leucine. And the fourth one is leucine. And then we're going to have GGG. GGG is glycine. Glycine is the next, and so on. So the messenger RNA sequence, the sequence of these triplets, determines the sequence of amino acids in the growing protein. One final thing about the genetic code here, you'll notice the redundancy in the code. So for example, CGG, CGA, CGC, and CGU all code for the same amino acid. However, if you have GAU, GAC, those 2 triplets code for this amino acid, but GAA and GAG specify a different amino acid. So sometimes changing the third position will change the amino acid, but oftentimes it doesn't. This is the redundancy. It's like for alanine here, we've got 4 triplets that specify alanine. And that's what we mean by redundancy. In addition, you should notice then that because of the redundancy, if there's a mutation in the DNA sequence that is affecting one of the letters in the third position of a triplet, see, then changing a G to a C won't change the amino acid. However, for this triplet here, GAA, if you change the third one from an A to a C, you are going to change the amino acid. When you go changing amino acids in a growing protein here, you might influence how it folds up, and therefore the function of the protein. If the change in letter occurs in the first position of a triplet, you are very likely going to be changing the amino acid. So changes to the first position will very likely change the amino acid, but changes to the third position oftentimes do not. change the amino acid due to this redundancy. Recall, there are 64 possible 3-letter triplets in the language of RNA and DNA, and only 20 amino acids. So we've got 64 names, in a sense, for 20 amino acids. Actually, we have less than 64 because, remember, 3 of these triplets are stop. So we have 61 names for 20 amino acids. The genetic code, then, is this relationship between the letters of DNA and messenger RNA, they're close cousins, and the language of proteins, which is amino acids. So this diagram is a translating device. It's like the Rosetta Stone, translating between two languages: the language of messenger RNA and the language of amino acids in proteins. And finally, it should be said that all living things are using this same genetic code. And this, of course, we've seen it before, is because this language was invented by the first life forms that then went on to give rise to all subsequent living things on Earth.